Ladies and gentlemen, there is a moment, often subtle and unannounced, that arrives in the lives of many individuals after the age of 50. It's a moment where the cognitive edge begins to dull ever so slightly. You forget a name you once repeated effortlessly, or you walk into a room with a purpose in mind, only to stand there blankly, grasping for a fleeting thought. You may dismiss it as just aging, but science tells us it's more than that. The truth is that our brains are extraordinarily plastic. They can regenerate, they can heal, and they can even rewire themselves in ways we once believed impossible. But just as certain behaviors and compounds promote this neuroplasticity, there are others that sabotage. What if I told you that one of the fruits you may have eaten your entire life, one often marketed as healthy and nutrient-rich, could be quietly undermining your brain's ability to repair itself after the age of 50? This isn't alarmism. It's not fear-mongering. This is hard neuroscience backed by biochemistry and molecular biology. Today, we're going to talk about this one fruit, but more importantly, we're going to explore the intricate biological pathways that get disrupted when you consume it. And we'll also look at how, by eliminating this from your diet, you may actually restore the natural mechanisms your brain relies on to maintain cognitive sharpness, emotional stability, and memory retention deep into your senior years. To understand how something as innocuous as a fruit could disrupt brain repair, we first have to appreciate what brain repair really means. The human brain has remarkable capabilities to regenerate. Neurogenesis, the birth of new neurons, continues throughout life, especially in regions like the hippocampus, which is critical for memory formation and emotional regulation. Then there's synaptic plasticity, the brain's ability to strengthen or weaken connections between neurons based on activity. This plasticity underlies our capacity to learn, to adapt, and to recover from injury. But here's the key, Liz. All of these regenerative processes rely on a delicate biochemical environment. Factors like brain-derived neurotrophic factor BDFN, insulin signaling, mitochondrial efficiency, and inflammatory cytokine levels determine whether your brain is in a state of growth and repair or in a state of stagnation and decline. Now, the fruit I'm referring to, though, commonly perceived as harmless, actively disrupts this balance. It interferes with BDNF signaling, promotes systemic inflammation, and worsens insulin resistance, all of which are direct antagonists of healthy brain function. That fruit is none other than the modern genetically modified banana. Yes, bananas. Let me be clear, this is not a vilification of bananas across the board. In their ancestral form, small, fibrous, and low in sugar, they were entirely different. But the bananas we consume today, particularly in Western countries, are hybridized versions loaded with high levels of glucose and fructose, minimal fiber, and an altered glycemic profile. And this matters deeply after the age of 50. So why does the banana become a problem after 50? It begins with insulin. As we age, our cells become increasingly resistant to insulin's effects. This is a natural part of aging, exacerbated by sedentary lifestyle and poor sleep. When we eat high glycemic foods like modern bananas, which can contain over 14 grams of sugar in a single serving, we provoke a strong insulin response. This creates spikes in blood glucose, which is followed by crashes. The fluctuation damages blood vessels, increases oxidative stress, and accelerates the decline of the blood-brain barrier, the very shield that protects your brain from toxins and pathogens. But insulin resistance doesn't just stop at blood sugar regulation. It impacts cognition directly. Insulin is necessary for optimal brain function. It modulates neurotransmitter levels and is required for the synthesis of ACE-line, which is essential for learning and memory. Studies have shown that insulin resistance is a major contributor to neurodegenerative diseases like Alzheimer's. In fact, some researchers now refer to Alzheimer's as type 3 diabetes. When you consume bananas regularly after the age of 50, especially without adequate protein or fat to buffer the glycemic load, you essentially train your body into deeper insulin resistance. This chronic condition creates a brain environment hostile to repair and regeneration. Then there's the fructose content. Modern bananas contain a roughly equal balance of glucose and fructose. Fructose, while naturally occurring, is metabolized very differently than glucose. It's primarily broken down in the liver, where it gets converted to fat through a process called de novo lipogenesis. The byproducts of this metabolism include uric acid and reactive oxygen species, both of which are neurotoxic in excess. Uric acid in particular is a silent antagonist of cognitive health. Elevated levels of uric acid are associated with reduced nitric oxide availability. Nitric oxide is a vasodilator that allows more blood and therefore more oxygen and nutrients to reach brain tissues. Without adequate nitric oxide signaling, brain perfusion declines. That means reduced oxygen delivery, impaired waste clearance, and slower neuronal recovery after stress or injury. Moreover, fructose metabolism depletes intracellular ATP stores. ATP is the energy currency of the cell. The brain, though only 2 of the body's mass, consumes more than 20 of the body's energy. Any interference with ATP availability translates into sluggish mental function, reduced neurogenesis, and impaired cognitive resilience. And we haven't even addressed the inflammatory cascade triggered by regular banana consumption in older adults. With age, the immune system becomes less efficient and more prone to chronic low-grade inflammation, a condition termed inflammaging. Bananas, due to their high sugar content and their specific fructose profile, elevate inflammatory markers like interleukin-6 tumor necrosis factor alpha F and C reactive protein ACO. This systemic inflammation doesn't stay in the body. It crosses into the brain, affecting microglial cells, which are the brain's resident immune sentinels. When activated, these cells can either support healing or destroy synapses and neurons. Chronic activation of microglia leads to cognitive decline, depression, and a reduced capacity for brain repair. Now, contrast that with what happens when these pathways are relieved of such burdens. When insulin signaling improves, when inflammation subsides, and when BDNF levels rise, the brain shifts into a state of regeneration. Neurons begin to grow new dendritic spines. Synaptic strength increases. Memory becomes more stable. Sleep becomes more restorative. Emotional resilience returns. All of these are signs that the brain is healing. We often look for complex solutions, supplements, pharmaceuticals, biohacking devices to stimulate brain repair. But sometimes the most profound shifts occur not by adding something, but by removing a key antagonist. When bananas are removed from the diets of individuals over 50, especially those already showing early signs of cognitive decline, the results can be startling. One study observed that over a period of 30 days, participants who substituted bananas with low glycemic fruits like berries experienced significant improvements in working memory and processing speed. Berries rich in anthocyanins and polyphenols promote neurogenesis and support BDNF expression rather than suppressing it. This simple switch yielded better sleep, more stable energy, and sharper mental performance. But I want to talk briefly about the mitochondrial connection here as well. Mitochondria, the energy-producing factories inside cells, are deeply involved in brain health. Dysfunctional mitochondria are implicated in nearly every neurodegenerative disorder. What drives mitochondrial dysfunction? High sugar diets, oxidative stress, and inflammation. The exact effects elicited by frequent banana consumption. When you reduce the glycemic burden on your mitochondria, you allow them to function more efficiently. They produce more ATP, they undergo proper autophagy, and they communicate better with the nucleus to regulate cellular repair. After the age of 50, your brain's resilience depends not just on what you feed it, but also on what you choose to withhold. Bananas, for all their historical reputation, simply do not support the biochemical reality of the aging brain. Their sugar content, their inflammatory potential, and their interference with key metabolic pathways place them in the category of foods best avoided if neuroprotection is your goal. And this isn't about demonizing a single fruit. It's about applying precision nutrition based on age, metabolic state, and neurological need. What worked for you at 25 doesn't work the same at 55 or 65. The rules of engagement change. The brain after 50 is more vulnerable to damage, but also highly responsive to the right stimuli. When we remove high glycemic fruits like bananas, we give the brain an opportunity to shift from a state of defense to one of growth. This opens the door to strategic interventions. Nutrients like omega-3s, magnesium threoate, and curcumin become more effective when they're not fighting upstream against chronic inflammation and glucose dysregulation. Ultimately, the decision to avoid this fruit isn't just about diet. It's about agency. It's about understanding that every choice we make, from what we eat to how we move and sleep, has a ripple effect that echoes through the brain's cellular architecture. 50. The stakes are higher. The margin for error narrows. But the potential for renewal remains enormous. The story of cognitive decline does not have to be written in stone. It can be rewritten deliberately, intelligently, and scientifically through targeted lifestyle changes. And sometimes it begins with something as deceptively simple as removing a familiar fruit from your plate. So let today be the start of a new chapter. One where the foods we choose are aligned with our biology, where we nourish our minds with intention, and where we no longer accept cognitive decline as an inevitability, but rather as a challenge we are fully equipped to meet head-on. Ladies and gentlemen, to truly understand the scale of the issue at hand, the silent creeping influence of a single fruit on cognitive longevity, we must dig even deeper into how our internal neurochemical landscape responds to the foods we eat after the 5th decade. It's not just about what a banana does in isolation, but rather how it interacts with the complex orchestra of hormonal, enzymatic, and neurological processes that are already shifting due to age. Consider the role of cortisol, our body's primary stress hormone. Cortisol is essential in small controlled bursts. It wakes us up in the morning, regulates inflammation, and helps us respond to acute stress. However, in the presence of a high glycemic diet, especially one with elevated fructose and glucose levels like that found in modern bananas, cortisol becomes dysregulated. Chronically elevated cortisol impairs hippocampal function, the very brain region responsible for consolidating memory and learning. Studies have shown that repeated exposure to high cortisol levels literally shrinks the hippocampus in both animal models and human subjects. Now imagine combining this hormonal stressor with an age-related reduction in sleep quality. Deep sleep, specifically slow-wave sleep, is the time during which the brain performs glymphatic cleansing, clearing away amyloid beta and other neurotoxic waste. But what promotes deep sleep? A stable blood sugar profile, a low inflammatory internal state, and robust melatonin production. Bananas disrupt all three. Their high sugar content leads to nocturnal blood sugar crashes, which in turn spike cortisol and adrenaline during the night, fragmenting your sleep architecture and denying your brain the cleansing it so desperately needs. Let's now talk about the microbiome, a hidden but potent regulator of brain function. After the age of 50, microbial diversity tends to decline. The species that remain can either be beneficial, supporting anti-inflammatory metabolites and neurotransmitter production, or harmful, producing endotoxins like lip polysaccharides (LLP) that damage the gut lining and lead to systemic inflammation. Bananas, due to their simple sugars and rapid fermentability, preferentially feed sugar-loving pathogenic strains of bacteria. This leads to dysbiosis, a microbial imbalance which has been shown to increase permeability in the gut lining, a condition often termed leaky gut. Why does that matter for the brain? Because the gut and brain are intimately connected via the vagus nerve and the systemic circulation of immune signals. When the gut becomes inflamed, it sends pro-inflammatory signals to the brain, particularly to the hypothalamus and limbic system, which regulate mood, appetite, and memory. People with leaky gut often report brain fog, poor concentration, and fatigue. And for good reason. The very chemicals produced in an inflamed gut reach and impair brain function. Now, if we shift from the gut to the vascular system, we see another major issue. The integrity of the vascular network that feeds the brain declines with age. Arteries stiffen, endothelial function worsens, and the ability to deliver oxygen-rich blood to regions like the prefrontal cortex diminishes. Glucose spikes from foods like bananas exacerbate this decline by creating glycation endoducts, molecular cross-links between proteins and sugars that stiffen blood vessels and impair their elasticity. These glycation end products also accumulate in the brain and are implicated in neurodegenerative conditions like Alzheimer's disease. The enzyme insulin-degrading enzyme ladder, which is responsible for clearing insulin after a meal, also happens to clear amyloid beta from the brain. When bananas are consumed regularly, especially in the insulin-resistant state, common after age 50, ID is overwhelmed with insulin and deprioritizes amyloid clearance. Over time, amyloid builds up silently, incrementally, and destructively within the brain. There's also the issue of dopamine sensitivity. Dopamine is a neurotransmitter critical for motivation, focus, and pleasure. After 50, the density of dopamine receptors declines, making it harder to feel reward and engagement from ordinary activities. Bananas cause rapid spikes in blood sugar, which temporarily boost dopamine, but then quickly crash it. This roller coaster trains the brain into a state of reward dysfunction. People begin to chase sugar-laden foods not for nourishment, but to stimulate an underactive dopaminergic system. This leads to a vicious cycle of overconsumption, followed by depression, phogus, and mental fatigue. And still the rabbit hole goes deeper. The aging brain's ability to regulate glutamate, a powerful excitatory neurotransmitter, becomes impaired. Too much glutamate leads to exotoxicity, damaging neurons and impairing synaptic plasticity. High fructose intake from bananas has been shown to exacerbate glutamate dysregulation by reducing the function of asterates, the brain cells responsible for clearing excess glutamate from the synaptic cleft. This damage is not just theoretical. MRI studies of older adults who maintain high sugar diets show measurable reducings in gray matter volume, especially in the frontal cortex and hippocampus. These are the regions involved in executive function, decision-making, and memory. Functional RRI reveals slower processing speeds, reduced working memory capacity, and poor attentional control in individuals consuming these high glycemic fruits daily. Most people eat bananas precisely because they believe them to be healthy. They're told that bananas are rich in potassium, which supports cardiovascular health, or that they provide energy, or that they're a natural source of sugar. But the form matters, the dose matters, and context, especially age, is everything. The potassium found in bananas can be obtained in higher quantities and more bioavailable forms from leafy greens, avocados, and even squash without the neurodegenerative liabilities. And yes, bananas offer some resistant starch when underripe, but how many people over 50 are consuming green bananas consistently? The vast majority are eating fully ripened spotted bananas, the kind that deliver a sugar load to your system nearly indistinguishable from a candy bar. It's time we rethink how we define healthy food, especially for the aging brain. It's not about how a food functions in isolation, but how it interacts with the evolving physiology of the human body as it ages. Foods that were once harmless, even beneficial, can become counterproductive as hormonal balances shift. Cellular defenses weaken and mitochondrial resilience wanes. Let's also talk briefly about mood. Depression and anxiety rates increase with age, and diet plays a significant role in this. The high sugar content in bananas influences the hypothalamic pituitary adrenal atriopaxis and exacerbates cortisol imbalances. Fructose also depletes magnesium, one of the most critical minerals for maintaining mood stability and neurotransmitter synthesis. Low magnesium impairs serotonin production and weakens the body's ability to buffer stress. Repeated banana consumption, therefore, doesn't just impair cognition. It also sets the stage for emotional volatility and mental fatigue. And return to our central theme, brain repair. What does brain repair truly require? It requires mitochondrial support, which means stable blood sugar and low oxidative stress. It requires BDNF, which is suppressed by inflammation and enhanced by polyphenols, exercise, and deep sleep. It requires adequate sleep architecture, which is disturbed by nocturnal hypoglycemia. It requires a healthy gut, which is thrown off balance by rapid sugar fermentation. And it requires emotional stability, which is eroded by dopamine spikes and crashes. When you eliminate bananas from your diet after age 50 and replace them with brain-supportive alternatives such as blueberries, which increase BDNF, or kiwi, which supports sleep via serotonin precursors, you begin to shift the brain's environment from degenerative to regenerative. You create a state in which the brain can grow new neurons, repair damaged pathways, and build resilience against future cognitive insults. It's easy to underestimate the impact of small daily decisions. One banana a day might not seem like much, but over the course of a month, that's over 400 grams of simple sugars delivered to a system that no longer processes sugar efficiently. That's 30 insulin spikes. That's 30 hits to your dopamine system. That's 30 events of low-grade inflammation. And over the course of a year, that becomes a systemic pattern, one that alters your brain's destiny. Ladies and gentlemen, neurogeneration is not a sudden cliff we fall off. It's a slow, quiet erosion. One banana at a time, one meal at a time, one night of fragmented sleep at a time. But the great news is that just as the decline is gradual, so too is the repair. The brain responds incredibly well to the removal of antagonists and the introduction of positive stimuli. Even people in their 70s and 80s have demonstrated neurogenesis when placed in the right environment. And so let us not be passive observers of our brain's trajectory. Let us be architects, engineers, scientists in our own lives. Let us design diets and lifestyles that support, not sabotage, our most precious organ. Let us replace the sweet comfort of bananas with the profound satisfaction of clarity, vitality, and cognitive endurance. Because after 50, every decision counts. Every meal counts, and every thought, word, and action becomes part of the narrative of our brain's future. Choose foods that write a story of strength, of wisdom, and of growth. Thank you.